Now again, in Chapter 6, we're trying to answer this question of whether or not cancer is something that can be inherited. And so we've discussed a little bit about DNA replication and how it is produced. But what we also wanted to look at in Chapter 6 is one form of cell division known as mitosis. And so in Section 6.3, that's on page 113, or begins on page 113 in your text, we're going to look at the 3 stages of the cell cycle. We're going to define mitosis and somatic cells. We're going to list the phases of interphase and describe the events, list and describe the phases of mitosis, and describe the process of cytokinesis. So as far as the cell cycle is concerned, the cell cycle is going to take place in 3 basic steps. First step is known as interphase, second mitosis, and the third cytokinesis. Now we're going to look at each of these in a little more detail, but just a couple of basic things to note about these at this point. So the main highlight of interphase is that this is where DNA is replicated. Now remember, we just talked through that process of DNA replication in the first section of Chapter 6. So all that we talked about there, with DNA copying itself, is going to occur in interphase. Then mitosis divides the nucleus. So all the other materials that are found in the nucleus, that's all going to be divided and into 2 different nuclei. Mitosis occurs in somatic cells or in body cells. So that term somatic just refers to body cells like skin, muscle, bone cells, those types of things. And then finally, cytokinesis is going to divide the cytoplasm. Now, if you remember, cytoplasm is all the contents of the cell, so the semi-fluid that sits in all the contents that are in it. And so if you divide the cytoplasm, essentially you divide the parent cell. So when all 3 of these steps are complete, where you started with 1 cell, you now have 2 cells that are exactly identical to each other. So let's look at these into a little more detail. So the first step of the cell cycle is interphase. Interphase itself has 3 phases, G1, S, G2, and they always occur in that order. G1, the first gap phase, is highlighted with 2 basic steps. So the first thing that's going to happen in G1 is the cell's going to grow. Well, keep in mind, we're getting ready to take this cell and split it in half. So the first thing you would want it to do is to grow, to mature to a level where it's actually able to split in half. And then secondly, we're going to see the organelles duplicate. Now remember, organelles, these are all those structures inside of a cell that carry out specific functions. So we learned these back in Unit 2. We talked about mitochondria and Golgi apparatus and ribosome. So all those different parts on the inside of the cell, they're going to duplicate during G1. So the question is, why would you want to duplicate all the parts of the cell? Well, again, keep in mind where we're going with this. We're taking 1 cell and we want to split it into 2 cells that are identical. Well, in order to be identical, that means they've both got to have mitochondria. They've both got to have Golgi. They've both got to have rough and smooth ER. And so all those structures are going to duplicate in G1. Now when the cell moves into the S phase, this is where the DNA replicates. So this is where that double helix is going to be split in half, and where DNA polymerase is going to come in and pair up new nucleotides with the original ones, following that rule, A with T and G with C. So at the end of S phase, all the DNA has been duplicated. And then remember, this is where it's going to, it's going to be held together by the centromere, making those identical sister chromatids. And then in G2, the second gap phase, this is where more growth is going to occur, and the cell is going to make proteins that are needed to finish out the cell division process. Now you want to keep it in mind that as far as the cell cycle is concerned, a cell is going to spend a majority of its time here in interphase. Because you think about it, it's going to take a long time to grow the cell, to duplicate all the organelles, to copy the DNA, and then to make all the proteins necessary for cell division. So majority of the cell's lifetime is going to be spent in the cell cycle. Now, once the cell cycle is, once interphase is complete, then the cell moves into mitosis. Now, again, we already noted mitosis is going to be division of the nucleus. Now, mitosis can only occur in eukaryotic cells. Why is that? Well, remember, eukaryotes have a nucleus. So if the definition of mitosis is to divide the nucleus, well, you've got to have one in order to divide it. Remember, prokaryotes do not have a nucleus, therefore they cannot go through mitosis. And again, the whole goal here is to take that one parent cell and to split it in half through this process. Now, if you flip over in your book, page 114 and 115, you will see the 4 major phases of mitosis. We collectively call it PMAT, P for prophase, M for metaphase, A for anaphase, and T for telophase. So what we want to do is to go through these 4 stages and get an idea of what's going on in the cell. So when the cell has completed interphase, it then moves into prophase, which is the first stage of mitosis. Now during prophase, some very specific things are going to happen inside the cell. First off, the chromosomes are going to condense. So they're going to condense into that normal X-shaped structure that we're used to seeing. And they're going to condense so that they don't become entangled as they move. Other things that we're going to see happen, the nuclear envelope that's surrounding the nucleus is going to disappear. Nucleolus is going to disappear, which basically means the nucleus itself disappears. And then we see microtubules, which are certain filaments. They're going to attach to the chromosomes and get ready to pull them around the cell. Now in animal cells, these microtubules or these filaments are going to be attached to centrioles at the different poles or sides of the cell. Plant cells, they don't have centrioles, but they still have microtubules present. So at the bottom of the page, you'll see the images located on the models in our lab. And so for prophase, we see that the cell actually begins here. So just get an idea, this is the cell, the purple bluish thing in the center, that's the nucleus. And so as the cell progresses through prophase, we'll see the chromosomes condensing into a more movable form, and we eventually see that the nucleus disappears. We also see these orange-shaped structures here that we would call the microtubules. In an animal cell, they're connected to centrioles at either side, and they're connecting to these chromosomes, getting ready to pull them around the cell. Now, once the cell has completed prophase, it then moves into metaphase. And those chromosomes are pulled by the microtubules to align them across the middle. So you'll notice, here's a centriole, here's all those fibers, or filaments, those microtubules. They're attached to the chromosomes and then move them to the middle. Now the question is, why would chromosomes need to move to the middle before the cell can divide? Well, remember, we want these two cells to be identical. So not only should they both have the same copies of all the organelles, but we want to make sure they get the same amount of DNA and that the DNA is identical. So by moving the chromosomes to the center, we ensure that the chromosomes split evenly so that whatever cell forms on this side has the exact same number and identical chromatids as what is found over here in this cell. Now, once the cell completes metaphase, it then moves into anaphase. Now remember, centromeres is what was holding those chromosomes together. So in anaphase, the centromeres split or dissolve, and the sister chromatids are pulled towards opposite poles. So you'll see here in this first image, this is where the centromeres that were holding these chromatids together, it separates. And then little by little, those microtubules start pulling these chromatids apart until they're found on either side of the cell. And you can notice that at the end of anaphase, you've got half of the chromatids on one side and their identical half on the other side. Then the cell moves into telophase. Now telophase, you'll notice that the nuclear envelope reforms around the two groups of chromatids. So if you look at the image on the top, so we'd have basically a nucleus that reforms up here around one half of those chromatids. And then at the bottom, you'll notice another nucleus forms around the other half of the chromatids. And then little by little, those chromosomes revert back to their uncondensed form and they become more thread or string-like. So here you have, bottom of page 114 and 115, this entire process. So we had interphase here, where it goes through G1, SG2. Then you've got prophase, metaphase, anaphase, telophase. Now be mindful that by the end of telophase, you still have just one cell, because remember, the purpose of mitosis is to divide the nucleus, not the cell. So at the end of mitosis, you have one cell that has two nuclei that are genetically identical to each other. In order to divide the cell has to go through cytokinesis. Remember, cytokinesis divides the cytoplasm. Again, it's happening after telophase, and it's going to split the cell into two new cells, or two daughter cells, that can then go through interphase and start the cell cycle over again. Now, cytokinesis is going to happen differently in animals and plants, mainly because of the outer barrier. Now, remember, for animals, we just have a plasma membrane around our cells. So what happens is a band of microfilaments is going to form in the center, and it's literally going to pinch the two cells in half, or pinch the cell in half. Plasma membrane is not a difficult thing to pinch. Now, for plant cells, remember, in addition to a plasma membrane, they have a cell wall on the outside, and you can't really just pinch a wall in half. So for plants, what happens in cytokinesis is they form what's called a cell plate in the center of the two nuclei, and this cell plate eventually develops cellulose and forms a new cell wall. So where animal cells pinch in half, a plant cell just forms a new cell wall. Now, I'm not going to play this animation for you, but I did just want you to kind of get an idea of what's going on. So for mitosis, remember, what's going to happen is you've got these sister chromatids that were held together at the center by the centromere. Remember, in prophase, they're going to condense, they're going to attach to these microtubules. So you can kind of get an idea. The nucleus, remember, is going to disappear. You can see them attach. When they move into metaphase, remember, this is where those microtubules are going to move them in alignment to the equator, to the middle. Then in anaphase, remember, the centromeres are going to dissolve, and the sisters are going to get pulled towards opposite sides here. And then in telophase, remember, those chromosomes are going to uncondense, and you're going to see a nucleus reform around each one of them. So by the end of this, you get one cell that has two identical nuclei. Then cytokinesis would take that cell. If this were an animal cell, it would pinch it in half, and you end up at the end of this with two cells that are genetically identical to each other.